So here's the thing that kind of broke my brain when I first read this. The same biological mechanism that could cure diabetes is like one wrong step away from causing cancer.
Yeah, it's the same switch, just how far you flip it.
Okay, so let's back up. We're talking about dedifferentiation, which is this idea that mature, specialized cells, cells that have a job, right? Like a pancreatic beta cell whose whole existence is to make insulin, can actually regress, like go backward developmentally.
Right. And the old thinking was that this is basically always bad. Cell loses its identity, stops doing its job, and you're looking at disease or malignancy.
But the newer picture is more complicated than that.
Way more complicated. Because it turns out cells do this as a survival strategy. Under stress, stress, oxidative stress, chronic inflammation, a beta cell will actually shed its insulin producing machinery to stay alive.
Which is, I mean, that's kind of remarkable when you think about it. The cell is basically saying, I cannot keep up with this workload and survive, so I'm going to become something simpler.
And here's where it gets interesting for therapy. If that regression is reversible, and the evidence suggests it often is, then you have a window, a therapeutic window where you can intervene and coax the cell back.
Right, but the catch is.
The catch is you can't go too far.
So, this is the Goldilocks problem the researchers keep running into. Too little regression, and you haven't actually rescued the cell. Too much, and you've essentially hit the reset button all the way back to a pluripotent state. Which, Alex, what actually happens then?
Teratomas. These are tumors that are genuinely bizarre. They can contain hair, teeth, multiple tissue types, because the cell has lost all identity constraints and is just proliferating without direction.
Which is what happens with full Yamanaka factor induction, the OSKM cocktail.
Exactly. OCT4, SOX2, KLF4, CMYC, those four factors are what Shinya Yamanaka used to first reprogram adult cells back to a stem-like state. Nobel Prize work. But in vivo, continuous expression is catastrophic.
So the question becomes, can you get the benefits of rolling back the clock without triggering that full collapse of identity?
And that's where DCAS9 comes in. This is the key distinction I want to make sure lands. Regular CRISPR-Cas9 cuts DNA. DCAS9, the D is for dead, catalytically inactive. It can't cut anything, but you can fuse it to other proteins, activators, repressors, things that write or erase chemical marks on histones.
So you're editing the epigenome, not the genome itself.
Right. You're not changing the sequence, you're changing which parts of the sequence are readable. And that distinction matters enormously for safety because you're not introducing permanent breaks that could be misrepaired.
Okay, so give me a concrete case. How does this actually play out in beta cells?
So the transcription factor PDX1 it's essentially the master regulator of beta cell identity. When a beta cell is under chronic stress, PDX1 expression drops. The cell starts expressing these dedifferentiation markers, ALDH1A3, CD81, stops making insulin. But if you use a DCAS9 VP64 system, VP64 is an activation domain, and you target it to the PDX1 promoter, you can reactivate PDX1 expression without touching the underlying DNA sequence.
And that's enough to pull the cell back?
In human islet models, it restored insulin secretion. It's not clinical yet, but the proof of concept is there.
Okay, but here's where it gets weird, and I want to spend a second on this: the math.
The damage importation problem.
Yes. So there are these mathematical models, PDEs, partial differential equations, tracking cells not by age, but by accumulated damage burden. And what they show is that when a heavily damaged cell dedifferentiates and rejoins the stem cell pool, it brings its damage with it.
It's like, okay, imagine you have a reservoir of clean water and you're filling it by recycling water from downstream, but the recycled water is contaminated. Over time, you corrupt the reservoir. Exactly. And empirically, dedifferentiation derived neural stem cells have shown exactly this. They carry functional damage that propagates forward.
So the therapy has to be precise enough to rescue cells that are early in the stress cascade, before the damage load gets too high.
which loops back to why the epigenomic approach, the non-cutting approach, is so important. You're trying to intervene at a checkpoint before the cell has crossed a damage threshold that makes regression unsafe. And then there's the cystic fibrosis angle, which is kind of a different mechanism, but it fits the same logic. Right, so the nanobody work is fascinating because it's operating at the protein level rather than the genomic level. The F508del mutation in CFTR, that's the chloride channel that's mutated in most cystic fibrosis patients, causes the protein to misfold in the ER. It never makes it to the cell surface. And ER stress from misfolded proteins is one of the exact triggers that purpose cells toward dedifferentiation. So if you can rescue the protein upstream, you might prevent the stress cascade from starting at all. The NB1R10 nanobody, developed at FMP and Charité in Berlin, actually enters the cytoplasm, finds the misfolded CFTR, and acts as a pharmacological chaperone, physically stabilizes it. Wait, how does a 15 kilodalton protein cross a cell membrane? 10 arginine residues, an R10 tag, positively charged, interacts with the negative charges on the cell surface, gets endocytosed, escapes into the cytoplasm. And combined with the existing triple therapy, elexacaftor, tezacaftor, ivacaftor, the combination pushed CFTR function to nearly 90% of normal in patient-derived airway cells. 90%? That's not treating symptoms. That's close to fixing the protein. In a dish, yes, but the directionality is clear. So where does this all land? What's the actual trajectory here? So Century Therapeutics has the CNTY813 program, iPSC-derived beta islets engineered to evade the immune system. They've knocked out HLA expression, so the host T cells don't recognize them, and they're overexpressing CD47, which is basically a don't eat me signal for NK cells. Off-the-shelf replacement beta cells that are essentially invisible to the immune system. IND application targeting 2026. If that works, and the preclinical data in diabetic mice is compelling, it would mean a functional cure for type 1 diabetes without lifetime immunosuppression, which is the part that gets lost in the technical discussion. There are people managing T1D right now who are doing finger sticks, calculating carbs, worrying about overnight roast. The biological infrastructure for a cure may already exist. The bottleneck is the safety question. Can you be precise enough with the cellular state manipulation that you stay in the therapeutic zone and don't cross into oncogenic territory? The Goldilocks zone. Yeah, not too hot, not too cold, just enough regression to restore function, not enough to lose identity. That's the episode. I'm Nova. And I'm Alex.
And this is Code and Cosmos,